The role of the 5-HT2A receptor in self and other initiated social interaction in lysergic acid diethylamide-induced states, a pharmacological FMRI study. Psychiatric disorders can be disastrous for a person's social interactions. It can cause problems with coworkers, friends, and even family, putting a strain on those relationships. Distortions of the self experience are especially noticeable in these disorders, and in order to improve this dysfunction, there is a need for further research on the specific neurochemical basis of these abnormalities in social interaction. Since it is difficult for an individual with a psychiatric disorder to coherently perceive their environment as it objectively is, it is not possible to understand the disease based off the patient's own subjective experiences alone, but rather their behaviors, social interactions, and neurobiology. One of the most promising techniques is pharmacological neuroimaging, to study the specific neurotransmitter systems within an individual while replicating a pharmacologically induced, altered state of consciousness. In order to achieve this internal state, the classic hallucinogen lysergic acid diethylamide, or LSD, is employed. This drug produces varying changes in the perceived self dependent on the dosage, and is an agonist at the 5-HT, 2AC, 1AB, 7, and dopamine 1 and 2 receptors. Since LSD is an agonist, pre-blocking the 5-HT2A receptor pharmacologically with a drug called ketanserin, an antihypertensive used to study the serotonin system historically, blocks the overall action of LSD, which gives reasons to investigate the specific system's relationship to the self and to social interaction. Of the behaviors that LSD alters, joint attention processing, which is the person's ability to understand another person's point of view, is one that is of most importance to our understanding of psychiatric disorders like schizophrenia, where it is generally impaired. By determining the specific systems at play within the brain during an experience, as well as analyzing eye gaze, we can understand the role of the 5-HT2R system better. With this background information in mind, our hypothesis is that 1. LSD loosens self-boundaries between oneself and other, altering one's perceived self-experience. 2. That LSD alters the joint attention processing of an individual by decreasing it both on neuronal and behavioral levels. And finally, that LSD is at least partially blocked by ketanserin. 24 participants gathered from a university were briefed on the experiments taking place. They were instructed to abstain from alcohol 24 hours in advance. In order to test the hypotheses, we used a gaze-based virtual game in which a subject's interaction with a virtual character was monitored and measured according to baseline levels. The subject's eye gaze was tracked and recorded for latency. Before, subjects received a 5D ASC questionnaire to measure the subjective effects of the treatment, which contained dozens of questions measuring one's perceived social expression. In a double-blind, randomized crossover design, there were 3 groups: one on the 100 micrograms of LSD alone, one on the 100 micrograms of LSD pre-treated with ketanserin, and finally, the control who just received a placebo. After FMRI data was collected as well as a statistical analysis performed. Ratings increased significantly on all 5D ASC questionnaires with LSD compared to the other 2 conditions except for spiritual experience and anxiety, which did not increase significantly for any group. After the drug administration, scores on both the positive and negative affect scales were significantly greater in the LSD treatment condition than in both the placebo and KET plus LSD treatment conditions. This meant that individuals with LSD alone were much more enthusiastic, but also much more neurotic. This slide represents what was explained in the previous. By looking at the red bars compared to the purple and green in Figure A, we can see a huge difference between the percent ratings of the individuals on LSD. In the graph 2, or B, we can see that the positive and negative affect were both increased significantly in individuals on LSD. Using the fMRI, we were able to find significant activity on both sides of the middle temporal gyrus, which is central for a number of cognitive processes. These include language and visual processing, as well as semantic memory processing. In short, this area is part of what makes someone's personality, what makes you, you. There were significantly higher bold signals detected in the left PCC on placebo versus LSD and in KET versus LSD when comparing self to other. Also, there were significantly higher bold signals detected in the medial prefrontal cortex on placebo versus LSD and KET versus LSD as well. Latency to establish eye contact was not significantly different between the drug conditions, and changes in mood were not significantly correlated with bold signals either. To put it simply, when on LSD, individuals had dampened activity in these areas. LSD was found to decrease the self-initiation of participation in the PCC and angular gyrus by the combination of neuroimaging using fMRI and using eye tracking in the visual game. Both of these brain areas are also supported by previous research on the topic of self-regulation and perception, as they were active in these studies as well. Altered connectivity and activity in these areas, while under the influence of LSD, is related to the dissolution of the self and is also consistent with previous studies. It was also found that LSD reduced self-related processing correlated with the activity in the associated brain areas, blurring the line between self and other, or in other words, dissolving the ego. For some psychiatric patients, this could be of use, while in others, maybe the opposite of what they need. From the results so far, it is clear that the serotonin system is not a simple one and may need to be altered in a particular way to improve in an individual's conditions. It was also shown that the 5-HT2R receptor has a critical role in the self because when it was blocked by cantharidin, the LSD-induced reduction of self-other differentiation was brought back to base levels to that placebo group level. Under LSD, we saw a reduced ability to establish joint connection with others, although not as dramatically as LSD's effects were on self-processing and subjective experience. This means that while our theory of mind may have been slightly diminished by the effects of LSD, our ability to connect with others through the dissolution of our own self was increased more heavily in this scenario. Overall, it is important to recognize that LSD loosens one's self boundaries by dampening the connection of self to other activity in the related brain areas of the PCC and angular gyrus specifically. LSD was able to reliably produce an unstable state of self-representation, similar to individuals with schizophrenia, as seen in social withdrawal behavior measured with eye gaze. From this, one might consider using a 5-HT2R antagonist to treat this specific disorder. For other disorders, like depression, that require an increased self-focus, a 5-HT2R agonist may be more appropriate. By using the graphs that were generated during this experiment, specific personality traits or ideas of the self can be modulated using this serotonin system. Further research of this pharmacology is important because it may hold the key to treating debilitating psychiatric disorders and understanding their origins. We measured LSD's ability to produce a state of lesser self-representation, but this is not the drug's only effects. It is important to also investigate the potential aspects of this drug in use for psychiatry, as well as its potential dangers. Following up on the participants would also be important in the following months because it may have led to lasting behavioral or personality changes that are worth noting. Overall, the experiment led to a clear conclusion. We do not yet know enough about the serotonin receptor system, but there is plenty of research to be done in the future, and possibly some exciting discoveries. A limitation of this study is the unequal gender distribution of 18 males and 6 females that were gathered from an ad at a university. With more men than women and a population of like-minded people, these individuals were not completely representative of all types of people that may benefit from research like this. This experiment could have benefited from a more diverse and possibly larger group of participants who were gathered as randomly as possible. Another fallacy of this study is that the other receptors stimulated by LSD may not have been investigated. We do not know all of the receptors responding to this specific pharmacology and how other receptors and systems may be at play during the testing and measurement phases of this study. Further research on the exact function of the molecule and receptor action would be beneficial for future research. However, it is important to realize that all other conditions were stable between groups, and the study remained valid. The results overall were good, and all 3 hypotheses remained true. They were not disproven. Finally, to expand the experiment, one other thing that could have been done would be to include a series of cognitive tests to test the overall thinking ability of each participant before and after the trials. This would create a new dimension that could easily be measured at the same time. Read the actual research here. Thank you.